Hello everybody, welcome to lecture 18. Today we're going to be covering gluconeogenesis and the regulation of glycolysis, as well as gluconeogenesis. So yeah, let's start. So, gluconeogenesis, I want to preface this by saying that gluconeogenesis is not just the reverse of glycolysis, and there is a slide below that kind of reiterates that, but gluconeogenesis, not the reverse of glycolysis, and where does it occur? So gluconeogenesis, for the cases of this class, you're going to say that it mainly occurs in the liver. It does occur in the kidneys as well, but for this class, we're thinking about the liver, right? So tissues that synthesize glucose is liver, and tissues that use the gluconeogenesis as their primary source, tissues that use glucose as their primary energy source are just like every other muscle in the body. Most of these other muscles do not actually perform gluconeogenesis. As far as I am aware, gluconeogenesis is specifically for the liver so that it can actually release glucose into the blood when you have low stores of glucose. So, how does it work? Gluconeogenesis is generally just the synthesis of glucose from things that are not carbohydrates. So you can use things like lactate, you can use things like amino acids, you could use triacylglycerols, you can use in plants you can use CO2 fixation, but in animals we're generally thinking about these guys here, right? So it's usually a cytosolic pathway as well, like because it's basically almost all occurs in the cytosol. It's kind of like glycolysis, but again, it's not just glycolysis; it's in reverse. It's a little bit different. And the energy and the carbons come from a lot of things. So fatty acid oxidation, which you'll cover in 441, lactate, amino acids, glycerol, all that jazz, right? And we have this thing, the Cori cycle, right? So it's what's funny about this whole process is, your muscles, right? When the oxygen is low, right, they'll do anaerobic respiration and produce a lot of lactate, right? Well, lactate goes into the blood, and actually that lactate goes into the liver cells, and the liver cells turn that back into glucose, and then shoot that back up into the muscles, so that you basically use more glucose. So things are recycled, things are used. Our lactate, right, came from glucose initially, right, and then you can take that lactate, basically turn it back into glucose, and then give it back to the muscle using the liver. The liver is a very powerful organ. And here we go. Gluconeogenesis is not the reverse of glycolysis. Now, these reactions look exactly the same, just reversed, but they are not, right? That is something we have to keep in mind, and I will show you how that works. So if you look here, the reverse of glycolysis would be this reaction. It would be very positive, but if we look at gluconeogenesis, it's actually quite negative. But it's energetically expensive, and it's an anabolic process, right? It's a building up process. And you can say it's energetically expensive because if you look at the reverse of glycolysis, right, this only uses 2 ATP, but this gluconeogenesis uses 4 ATP and 2 GTP. So basically 6 TP equivalent. So 3 times the amount of TP energy in comparison to just the reverse of glycolysis, right? That's how you actually get this. It's actually the way that you're actually able to get this negative delta G values because of the fact we're using 6 TP molecules to actually drive favorability. So these are the 3 steps, right, that if you remember are the main negative steps in glycolysis: step 1, step 3, and step 10, right? These are the main negative steps, right? So we got to figure out here, if that's the case, how does that work for gluconeogenesis, right? Because if they're super negative in one direction, they're not just going to go back up the same direction super easily with just the same enzyme. So actually we have different enzymes. So for step 10, we have pep carboxykinase and pyruvate carboxylase. For step 3, we have fructose 1,6 bisphosphatase. And then for step 1, we have glucose 6-phosphatase. So phosphatases, they remove phosphate groups, which makes sense, right? If a kinase adds a phosphatase, right, then a phosphatase removes it, right? Step 10 is a bit weird because basically what we do is we convert pyruvate into pyruvate carboxylate, turn it into oxaloacetate. And if you remember, if we're adding a carboxyl group, I talked about this in the previous lecture, you're going to be using biotin, which is vitamin B7 most likely, right? And then you also have PepCK, which is a separate reaction that basically turns it back into phosphenolpyruvate. But we'll get into that in a second. So if you look at this, starting from step 10, right, we're going to look at this, right? So pyruvate carboxylase, right, is the main is one of the main enzymes in this kind of reverse process, and as you can see, it uses vitamin B7, right? Because it's a carboxylase reaction. So like I don't even have to know that it uses vitamin B7 because it's been a while since I covered this material to know that it uses vitamin B7 because I know it's a carboxylase reaction. Therefore, I'm immediately thinking that it probably uses a biotin as one of its mechanisms, right? So if you look at this, right, pyruvate carboxylase, all we're doing is we're taking this bicarb and we're adding it here, right? Basically some sort of enolate mechanism, right? And we're using biotin, right, to turn it into oxaloacetate. And you will see that this is actually an intermediate in the citric acid cycle when you eventually cover that for the next exam. So if we look at how this works, the way this actually works is, we need ATP. So why do we need the ATP? We use the ATP to activate this bicarbonate to then add it on to pyruvate carboxylase. So by activating this bicarb and turning it into this carboxyphosphate, right, we are able to then attach it to biotin, right? Attach this carboxyl group to biotin, right? This carboxyl group to biotin, right? And you don't have to know the exact mechanism. And then that biotin basically acts as a swinging arm. It basically you can think of it as like a lever, right? It basically goes from one place to another, right? And it basically by moving to site 2, so at site 1, right, it does this whole reaction here. At site 2, it does this separate transferase reaction, right, where it takes pyruvate in the enol form, right, and then it forms this oxaloacetate molecule. The second step is PEP carboxykinase, right? So once we have formed this oxaloacetate, right, from pyruvate carboxylase using ATP and biotin as a cofactor, right? Biotin, right? We can then think about what we're doing from here. So after that, right, what are we doing? Well, from here, right, we have to go from oxaloacetate to phosphenolpyruvate. And the way this is done is we have to use a GTP molecule. And this is, again, one of those a little bit bizarre mechanisms, but basically what's happening is that we're actually going to release this CO2 that we just put on there, and that's going to be very entropically favorable, right? So if you remember entropy, right, because we're releasing a gas, gases are very entropically favorable to release. So basically, we're getting rid of this CO2 here, and then we're using that energy to bring these electrons back to this bond, and then this will grab a phosphate group, right, from GTP, right? So we're using the release of CO2, very entropically favorable reaction, to grab a phosphate group from guanosine triphosphate. And the breakage of guanosine triphosphate is also very negative, generally speaking. The addition of a phosphate group to any molecule in general is not the most favorable, but we have two reactions here that are driving this reaction of to turn it into phosphenolpyruvate forward, right? So basically, right, the main gist here is that in step 1 with pyruvate carboxylase, we're using ATP and biotin, right, and bicarb as well to add ATP, bicarb, and ATP, bicarb, and biotin, right, to add this CO2 group on, right, to the biotin, and that biotin acts as a swinging arm, and that CO2 that was formerly came from the bicarb is now being added to so that CO2, right, is then being added to this pyruvate, right, to make oxaloacetate, right? That makes sense, right? So pyruvate, right, bicarb, ATP, and biotin, right? Those are the main things you need for this reaction, right? You need the bicarb, you need the ATP, you need the biotin, and then you need the pyruvate. I want to stress this, right, because they will most likely ask you questions about this, right? And remember what they release and what they absorb, right? So this actually technically does not use CO2. It releases CO2, but it actually technically uses bicarb, right? So this actually releases CO2 in the process after this step here, right? But something to keep in mind. In the second step of this reaction, right, returning oxaloacetate into phosphatidylpyruvate, and this uses GTP and releases CO2, right? So there's actually a lot of things, and this is catalyzed by PEP carboxykinase, right? So there's actually a lot of things to keep in mind here, right? But the main things you should keep in mind are what is being used, so like, and what's being produced. So like this, like the bicarb, this ATP, right? The biotin, the pyruvate, of course, what happens there? The biotin acts as a moving arm, attaches the bicarb onto it, right? And then we get this oxaloacetate. And this oxaloacetate combined with GTP does this carboxykinase reaction where CO2 is released, right? And GDP is formed, and you get phosphatidylpyruvate. So pyruvate carboxylase, the main reaction, the main enzyme for this guy here, and PEP carboxykinase is the way that we release here. So, from PEP, phosphatidylpyruvate, to fructose 1,6-bisphosphate, this is pretty much just the reverse reaction of glycolysis, except it uses a different enzyme, right? So, you recognize that, for this, fructose 6-phosphate to fructose 1,6-bisphosphate uses fructose phosphofructokinase 1, right? Which is just a kinase. Well, fructose 1,6-bisphosphatase is just a phosphatase that removes the phosphate off of it. That's all. There's nothing else to it, and it's still favorable, right? Still favorable. And similarly, if you go up here to this up here, you got hexokinase or glucokinase in the liver, right? And the one that removes it is actually glucose 6-phosphatase, right? And what you should know about this glucose 6-phosphatase, right, is that it is only present in organs that make glucose or release it into the blood. So, for example, your liver and your kidneys have this glucose 6-phosphatase, but your muscle cells do not, because they're not going to be making glucose to release it into the blood. Why would they do that? They need to use the glucose themselves, right? So the muscles do not have glucose 6-phosphatase, right? The liver does, though. So, we've got this thing about steady state flux. I don't think this is the most important thing in the world. I think the main thing you should keep in mind from this is, like, every reaction has like a different velocity here, right? But the rates are all the same, even though like the actual like speeds are not. The rates look the same because the net rate is the same, right? But the net difference between the forward and reverse reactions are actually the same, despite the overall reactions, like the individual reactions themselves, probably taking different speeds. So this is how do you regulate enzymes, right? There's a whole bunch of ways you can regulate enzymes, right? And here are some examples. Hormonal, you'll cover with insulin glucagon. You got compartmentalization. You've got post-translational modifications like phosphorylating something, or like allosteric control, like those heterotropic or homotropic inhibitors, right? Like, I think we've talked about that, or like modifiers, right? Transcriptional translational regulation. Is a protein transcribed and translated? Is it not? There's a whole bunch of things you got to keep in mind for this, right? And the question is, how do you know which enzymes are regulated? And I told you that I know that steps 1, 3, and 10 are very regulated, right? Well, why do I know that? Well, I know that because these 3 steps are very, very negative in delta G, right? It's basically like the points of no return, right? So you basically want to regulate it before those steps happen, because once they happen, you're not going back, right? And so the point is, if you see if they give you a made up pathway, right, and you see a very negative drop in delta G, right, that's how you know that step is most likely reversible, and that step is most likely regulated, right? So Now, these are very important. You must know these. These are some of the hormones that are important in metabolism, and you have to know how these work, because it makes a very big difference. So insulin, right, and glucagon are the two main ones, and then epinephrine is a bit of a fun one. But um, glucagon, right, basically, if you're in the starving state, right, you need to satisfy the natural level of blood glucose required to be a functioning being, right? And the way that your body does that is by releasing glucagon. And this signal actually tells the liver to actually release sugar into the bloodstream so that your other organs can pick it up, basically. Insulin is the opposite of that. Insulin is after you've eaten a meal, you have a bunch of sugar in your system. Insulin is released to insulin's released to actually store sugar in both your muscle and your liver, right? Because your muscles can actually store glycogen as well, right? But it's released as a signal to store sugar, right? And these are released by the pancreas, right? So your liver is a separate thing from the pancreas, right? Your pancreas is responsible for the hormones, but then those hormones actually act on the liver, right, to actually cause some sort of response, right? But the pancreas, I think I might have said the liver releases them earlier. That's a mistake. But basically, the liver it responds to these hormones as well as any other all the other tissue in the body. And epinephrine is basically similar to glucagon. It is actually an amine hormone. It's an amine hormone because it's got an NH here. It's released by the adrenal medulla. There's actually two versions. There's two types of things in the medulla. You don't have to know this, but the kidney actually has the adrenal cortex and the adrenal medulla. It's separated into two parts. The adrenal medulla is the innermost part of it, and the adrenal cortex is the outermost part. And the adrenal cortex is actually responsible for synthesizing things like corticosteroids. So things like cortisol, for example, is synthesized by the cortex of the kidney, but or the adrenal, of the adrenal, right? Sorry, of the adrenal gland. Kidney is not the same as the adrenal gland. Anyways, that's not too important, I don't think, for your exam. Just a cool little fact to know. But if we look at this, right? Epinephrine is pretty much the same as glucagon. Like, if you're fight or flight, if you remember this from Bio 220, right? If you are in fight or flight mode, you're going to want a lot of glucose in your blood, right? And so epinephrine triggers that release of glucose concentration into the blood, and then that gets absorbed by your tissue, and then you start running away, right? So to summarize, glucagon stimulates the release of glucose into the blood, insulin triggers the storage of glucose, and epinephrine also triggers the release of glucose into the blood. These are all released. These two are released by the pancreas, and this is released by the adrenal medulla. And this one, I don't think you have to know that it's released by the adrenal medulla, but you do have to know what epinephrine is. So, if we look at the regulation types, we are looking. This is just a nice little graph of if we're looking at hexokinase in the liver versus the muscle. You can see that the Km, right, is much higher here than it is here, which means that the muscles require much less glucose to reach half Vmax, right? But notice the Vmax is actually kind of lower, so this is something to keep in mind. I don't think it's too important, but there's other things to focus on here. So, did we talk about regulation of? I guess we're just going here. Interesting. They kind of just skipped over glucose 6-phosphate a little bit. I'll talk about it later because I think it's a little bit important. Basically, okay, regulation of phosphofructokinase 1 of PFK1. Well, these should actually make intuitive, except for this one. This one's a bit weird, but these should actually make a lot of intuitive sense, right? So, if we don't want to do glycolysis, we don't want to go this way, right? How do we inhibit it? Well, the products of glycolysis are what? Generally speaking, ATP, pyruvate, all that jazz, right? Now, if we're producing a lot of ATP and pyruvate and citric acid cycle products, we're not going to want to make more of them, right? So you would probably say, okay, well, let's just make sure that PFK1 is inhibited by things like ATP, pyruvate, citrate, or citric acid cycle intermediates and stuff like that. And that's actually what happens, right? ATP, product of glycolysis. Citrate is actually a intermediate in citric acid cycle. So you can imagine if you have a lot of citrate, you're doing a lot of citric acid cycles. So you don't need more glycolysis to happen, right? So because we don't need more glycolysis to happen, we can just use the citrate that we make in citric acid cycle to go back and inhibit glycolysis, right? On the other hand, if we have low levels of ATP, we're going to have higher levels of ADP and AMP, and those are going to act as activators, which makes sense, right? So we're going to have ADP and AMP be activators of it. Fructose 2,6-bisphosphate is I think is explained a little bit later, but this is actually a side reaction. So instead of doing fructose 6-phosphate into fructose 1,6-bisphosphate, if you have too much fructose 6-phosphate, some of it will actually be turned into fructose 2,6-bisphosphate as well as fructose 1,6-bisphosphate. And this fructose 2,6-bisphosphate actually acts as a supercharger for PFK1. Basically, it actually activates that enzyme because there's so much of it that you're actually able to make fructose 2,6-bisphosphate. It tells PFK1 that it needs to start working harder to lower that concentration of fructose 6-phosphate. And here is an example of what that enzyme looks like. You don't need to know how the structure looks. It's a bit of a hassle. So if you look at the inhibition here, here's what it looks like. If you look at PFK activity versus fructose 6-phosphate, you can see that with low ATP, right? ATP is an inhibitor of PFK1, right? Because PFK1 promotes glycolysis, and ATP is a product of glycolysis. So ATP must inhibit PFK1 because we don't want to make more ATP, right? Hopefully that logic makes sense, right? You can see that it reaches this Vmax a little bit sooner, a lot sooner actually, generally speaking, than when it would be reached if there was high ATP, right? And then the Km is much higher with high concentration. So if we look at the 50% points around here, right? So with low ATP, it's there, and around high ATP, it's over here, right? So clearly, ATP has a pretty dang big effect on it. Now we talk about the holy grail, fructose 2,6-bisphosphate. So fructose 2,6-bisphosphate is a very important activator of PFK1, and it substantially lowers the Km, so it increases the affinity of PFK1 for free fructose 6-phosphate. Makes sense. Don't really see a problem with that, right? So what is happening? Basically, right, this fructose 2,6-bisphosphate, when added, right, will allow the Km to be around here, but if there's no fructose 2,6-bisphosphate, it's all the way over here. Notice that it's a massive difference in concentration required to reach half Vmax. Like, actually massive. It's like this is way bigger. I can't do that. Like 40 times larger, right? If there's no fructose 2,6-bisphosphate. So it's a very potent activator of PFK1. Right? And it has a reciprocal regulation for glycolysis and gluconeogenesis, right? So fructose 2,6-bisphosphate, right, activates glycolysis, it activates PFK1, but it also inhibits fructose 1,6-bisphosphatase, or FBPase, right? So it activates PFK1, phosphofructokinase 1, but inhibits FBPase, fructose 1,6-bisphosphatase. And actually all of these inhibitors and activators do that. So ATP, for example, will activate FBPase but inhibit PFK1. AMP, for example, will activate PFK1 and inhibit FBPase, right? Basically, it depends on whether or not it's a product or a reactant of glycolysis, or like if it signifies high or low energy states, right? And AMP signifies low energy state. It'll activate PFK1 to produce more energy and inhibit FBPase to stop it from trying to counteract that energy formation, right? And all these inhibitions kind of and activations kind of counteract each other to get this complex system of regulation, right? And here, right, we're looking at FBPase, and it should be, if it's an activator, right, of PFK1, right, it should be an inhibitor of FBPase. And you can see that here, right? When you look at FBPase activity, right, it should be inhibited, right, with fructose 2,6-bisphosphate. And it is. If you look at the KM here, without fructose 2,6-bisphosphate, FBPase is very active. But when you add it, it goes way down. It goes from like 0, like 0 point something, to like 75 or 90, right? It's so much larger when fructose 2,6-bisphosphate is added, right? Just goes to show that these are very, very important inhibitors, and it's a very important molecule for the regulation of PFK1, right? And here is a example of how this works, right? So here's how it works, right? If you're in a very fed state, you're gonna have a lot of ATP, right? You're gonna have a ton of it, right? And if you're gonna have a ton of ATP, and you have a ton of glucose, right? Some of that glucose is gonna get turned into to glucose 6-phosphate, isomerized into fructose 6-phosphate, and then a lot of that fructose 6-phosphate will go and be turned into phosphofructokinase. Sorry, it'll be turned into fructose 1,6-bisphosphate, right? But some of it will go through PFK2 and be turned into fructose 2,6-bisphosphate, right? And this fructose 2,6-bisphosphate will be used to supercharge and increase the affinity of PFK1 for fructose 6-phosphate to turn more of that fructose 6-phosphate into fructose 1,6-bisphosphate, right? And there's another example of it here, actually. But, that's the interesting part. And then also, that, fructose 2,6-bisphosphate, this can be turned back into fructose 6-phosphate using FBPase2, right? Which is a fructose 2,6-bisphosphatase, right? And, that is usually what happens in the starved state, right? And, here we go. It's regulated by blood glucose levels, and you can see, all of these things that, we've talked about, right? And, the thing you should actually recognize is these are, like, actually, like, kind of, they're, like, one enzyme complex, kind of. So, fructose 2,6-bisphosphatase is actually kind of combined with fructose 2,6-phosphofructokinase 2, right? So, like we said before, fructose 2,6-bisphosphate stimulates glycolysis, and inhibits gluconeogenesis, right? Lower fructose 2,6-bisphosphate inhibits glycolysis and stimulates gluconeogenesis, right? And, if there's one thing I want to say about the hormones, right, it's glucagon and insulin, right? So glucagon, I'm going to write this actually down. So glucagon, right, equals phosphorylation, and insulin equals dephosphorylation. You can actually use this principle to actually kind of inform what you expect to happen. So, for example, right, I know that glucagon, right, is released when energy stores are low, right? And so if glucagon is released when energy stores are low, that means we're going to want to make more glucose, right, in the liver. So let's think about how that works. That works, right, because if we have glucose, right, if we have glucagon, right, that means we're in a starved state. So what we're going to do is we're going to phosphorylate this enzyme at this position here. And since we want to make glucose, we want to turn off PFK2 and activate FBPase2, right? And see, if we look at that, glucagon, right, puts ATP on this, phosphorylates this, and the FBPase2 becomes active and the PFK2 becomes inactive, right? On the other hand, right, insulin, right, is a dephosphorylator, right? And we're starting at this position, right? And insulin suggests that, the body is, has a lot of glucose and it needs to start storing, right? In that case, if you look at this, insulin dephosphorylates, it would dephosphorylate this, get rid of it, right? It would activate this kind of phosphatase, right? And it would turn it into this form, right? Where now the PFK2 is active, right? So you can basically use these rules here to kind of guide your guide your intuition or thinking onto whether or not an enzyme is going to be dephosphorylated or phosphorylated in its active state, right? Because if you look at this, right, the FBPase is active in a phosphorylated state while PFK2 is inactive in a phosphorylated state, and it's the reverse for for PFK2, it's active without the phosphate, and for FPPase, it's inactive without the phosphate. So it kind of changes, right, depending on what the enzyme is and what process it's contributing to. So something you need to keep in mind, right? And here's some proteins you should keep in mind as well. So for PFK, for this kind of fructose 2,6 bisphosphate complex, right? The enzymes that are responsible for this phosphatase is phosphoprotein phosphatase, so PPP, and then cyclic AMP dependent protein kinase, right? Glucagon, right, leads to a signal cascade with cyclic AMP, which leads to phosphorylation through CAMP dependent protein kinase, right? And insulin leads to the activation of phosphoprotein phosphatase. So generally speaking, insulin leads to the activation of phosphatases, and glucagon leads to the activation of kinases. That's something to keep in mind, right? Go ahead and add that here. Equals kinases. Oh god. Equals kinases. Okay, right done. Equals kinases. And insulin equals phosphatase. There you go. So this is generally the rule of thumb you can use for these reactions. Oh, dear goodness. Right, and this, right, will pretty much get you through all of those reactions, right? Use this, right, and you will figure out how pretty much all of these pathways work. Now, pyruvate kinase, is much of the same, right? There's really no secret here. We're back to the protein phosphatase, right? So, if we look at pyruvate kinase, right? And of course, gluconeogenesis only occurs in the liver, right? But pyruvate kinase, right? Well, let's think about this, right? If we have a lot of glucose, right, we're gonna want to do glycolysis, right? And insulin promotes storage, right, and the performance of glycolysis, right? And insulin leads to dephosphorylation, right? And we expect that in that case, if we want to activate it, right, we're gonna actually use insulin to use our phosphatase, or our protein phosphatase, to activate this enzyme. Yeah, voila! It stimulates glycolysis when in the dephosphorylated form. Right? Let's look at glucagon instead. Glucagon is for when we're in a starving condition, and we're not going to want to, do glycolysis in that state, right? So glucagon is going to lead to phosphorylation of pyruvate kinase, right? Which leads to the inactivation of pyruvate kinase, right? Using our protein kinase A, right? So you can kind of see, right, how we're using that general rule of thumb to kind of see how this works. So, if you look at glycolytic tissue, right, for pyruvate kinase, it's the same things that you've seen before, right? How do we stop pyruvate from being formed, and how do we stop glycolysis? Well, if the products of glycolysis and the downstream cycles go back and inhibit glycolysis, then we're not going to make any more of it. So ATP is a great example of it. Acetyl-CoA is actually created by pyruvate dehydrogenase. It actually uses pyruvate to make acetyl-CoA, right? So that makes sense, right? If acetyl-CoA is actually farther down in the pathway, acetyl-CoA will come back and inhibit it. Long chain fatty acids, right, are basically just it's just a bunch of acetyl-CoAs combined together, right? So if you can imagine, if we just have a bunch of acetyl-CoAs, right, it's going to inhibit the formation of more pyruvate, right? Since it's like feedback inhibition, right? And then alanine is actually just a transamination reaction of pyruvate. You don't have to know, the insides and outs of how this works, but if I draw it here for you, right, try and draw what this looks like. You draw out the structure of alanine. This is the structure of alanine, right? And what I want you to notice, right, is that the only difference between these two is this group. And what you'll learn in Biochem 441 is that there are enzymes called transaminases. And these transaminases literally take a carbonyl group and they switch it with an amine group. And so what happens is you get these two groups. You can basically just take pyruvate, switch that carbonyl group with an amine group, and you end up with alanine. And so alanine, right, is now an inhibitor of pyruvate kinase. Right? So you can see here, right, alanine, ATP, acetyl-CoA, these long-chain fatty acids, these all make sense. These are all products or downstream products of the glycolysis reactions, and these come back and inhibit glycolysis. The other thing to keep in mind is that fructose 1,6-bisphosphate, which is a previous intermediate in glycolysis, acts as an activator for pyruvate kinase, which makes sense, right? Because if we have a lot of fructose 1,6-bisphosphate, we're going to want to tell the rest of our enzymes, especially pyruvate kinase, to keep working at maximum capacity, right? Because if we have a lot of fructose 1,6-bisphosphate, that means that we need to keep doing glycolysis in order to just kind of deplete this excess amount of glucose that we have available to us, right? So hopefully you can start to see like some intuitive patterns here with what is considered an activator and what's considered an inhibitor here, and how that is actually very dependent on what enzyme you're looking at, right? So I hopefully that makes some sense, right? Because it's always important to try and take into perspective what each thing means. Like for example, what is ATP? An energy source, and it's produced by glycolysis. So if it's produced by glycolysis, it probably negatively impacts glycolysis if it were to go back and have an effect on glycolysis. And with acetyl-CoA, right? It's a product of glycolysis after you do pyruvate dehydrogenase, which you guys haven't covered yet, right? And so if it's a product of that, then what's going to happen after is we're going to use that acetyl-CoA, right, to go back and inhibit glycolysis, because we don't need any more acetyl-CoA if we already have a bunch of it. Same thing with long-chain fatty acids. They're just a bunch of acetyl-CoAs combined together, right? If we already have a lot of acetyl-CoA, we don't need to do more glycolysis, so we go back and inhibit it, right? You should be able to pick up on these patterns, right, of, what things mean, right, and how do they work. And of course, if you have questions, you can leave them in the comments or ask me at the IC. I'm more than happy to answer. These chapters and these lectures are just a lot of memorizing, so just important that you are vigilant and you are giving these giving these topics the respect they deserve, because they take a lot of work to kind of get in your head. And here is a nice little diagram of how all of this looks, right? So if you look at high blood glucose, right, we're going to have an active pyruvate kinase, right? You're going to have a lot of fatty acids, amino acid synthesis, and all that jazz. We're going to have a lot of protein phosphatase activity because insulin, right? Insulin stimulates dephosphorylation, right? And, we're going to have a lot of kinase domains. The PFK2 domain is going to be active, right? The fructose 2,6-bisphosphate is going to be inactive. We're going to have a lot of fructose 2,6-bisphosphate. Haven't talked about glycogen yet, but just think of it like glycogen storage, right? If you have a lot of glucose, right, you're going to want to store a lot of glucose. If you don't have a lot of glucose, you're going to want to release glucose from glycogen. So if you have a lot of glucose, we're going to store glycogen, right? We're not going to release it, right? On the other hand, if you have low blood glucose, right, you're going to have a lot of pyruvate kinase being inhibited, right? You don't want to do glycolysis. You're going to have a lot of fatty acid breakdown to make acetyl-CoA, which again, you know. Glucagon, right, is what's released with low blood glucose, and glucagon leads to phosphorylation, right, with kinases. And PK active, protein kinase A, right? And we're going to have a lot less fructose 2,6-bisphosphatase. We're going to lead to a lot more glycogen breakdown and less glycogen synthesis, right? So, yeah, so which reactions are different between glycolysis and gluconeogenesis? It would be step 1, 3, and 10, right? Which steps are in glycolysis are regulated? Also steps 1, 3, and 10. Explain the regulation mechanisms for regulated steps, right? This would be that the products of glycolysis and downstream pathways will come back and inhibit, these steps. So ATP citrate, alanine, these things will inhibit it. Things like ADP and AMP will activate these enzymes, right? Just as a way of saying, hey, we need more glucose, or hey, we don't need more glucose. And reciprocal regulation is this idea that one thing, or like one molecule, has opposite effects on different enzymes. So if we think about fructose 2,6-bisphosphatase and fructose 2 PFK2, right? Those are enzymes that are kind of combined together, right? And we think about how that's regulated. You can think about how, if you look at insulin, right? Insulin leads to dephosphorylation, right? Which leads to the activation of PFK2, right? And it leads to the inactivation of fructose 2,6-bisphosphatase, right? You can see that reciprocal regulation there, right? So that insulin activates one of those enzymes but deactivates the other, right? And that's kind of the idea of reciprocal regulation, is that when one molecule basically inhibits and activates two different enzymes, basically, that are complementary to each other. And, be familiar with well-fed versus starved state. So you should know how insulin glucagon work. Insulin, fed state, glucagon, starved state, right? What enzymes do they activate? Does it phosphorylate or dephosphorylate, right? How does that change? How does the phosphorylation or dephosphorylation affect the enzyme? Does it depend on what enzyme you're looking at, right? How is it different if you were to phosphorylate glucose 6-phosphatase versus pyruvate kinase, right? You should be able to think about these and be able to solve those in your head, and then go from there. The one other thing I want to mention is I don't really think they talked about this, but glucose 6-phosphate, so the hexokinase reaction, right? It's poorly drawn, but the glucose 6-phosphate actually goes back and inhibits this, right? So it's another way of another mechanism of feedback inhibition, right? The glucose 6-phosphate inhibits hexokinase, right? And then the other thing I want to mention is that, I think I said this before, but glucose 6-phosphatase is not in the skeletal muscle, right? So the other thing I want to say is that the fructose 2,6-bisphosphate pathway, so that PFK2 and fructose 2,6-bisphosphatase, those are only in the liver. So those do not occur anywhere else. So that's something to keep in mind. But generally speaking, that's like the major site of expression for this class. I think it's expressed elsewhere, actually, like I think maybe the heart and the kidney, for example, but I think for this class, it's okay to think about it as it's being only expressed in the liver. So, let me just double check that. Never mind, I lied. So yeah, though there is a version of PFK2. It looks like muscles, but I don't think that was mentioned here, which is why I was looking for it. Yeah, I don't really, they don't really mention it anywhere. I guess but in this case they're specifically using liver as. I would just say that it's okay to think of fructose 2,6-bisphosphate and that pathway being present in the muscles as well. It's generally that pathway is more prominent in the liver, but it does occur in the muscle cells as well. So yeah. Anyways, I hope that this video was helpful, and that a lot of confusions were cleared up. But if you have any questions, feel free to leave a comment or stop by the IC. I've got a workshop on Monday going over my exam from last year, and yeah, hope to see you guys there. Happy studying.